Today we're going to look at how to study some of Mendel's genetic studies using Punnett squares to determine probability. And so the procedure we want to follow goes like this. We start with the parent generation, and we will indicate parent generation as the letter P, and we want to then lay out the genotype of the parents. The parents both carry alleles from each of their parents. So for example, we started with tall plants indicated by capital T, which is dominant trait. So uppercase letter is going to be the dominant trait, uppercase letter, dominant, and he crosses homozygous or purebred tall plant with a homozygous recessive or purebred short plant. So lowercase letter indicates the recessive trait. Make a note here one time. Lowercase letter is a recessive trait. We need to then separate the alleles into the gametes. The gametes are the sex cells. So gametes are the sex cells, egg and sperm. And only carry one allele. For a given trait. So in our example here, we have this parent which only has one type of allele, and so the gamete, there's only one possibility. And for the short parent, we have the recessive allele, and that's indicated by the lowercase letter. So in order to make a Punnett square, we need to identify the number of boxes we need in our square. So in this very simple case, because there's only one allele variation, we have 100% of the offspring being what are called heterozygous or hybrid, and so this is called the first filial generation for F1. And so because this first generation of offspring will all have this heterozygous or hybrid where they have one allele from one parent that is uppercase and one allele that is lowercase, we say that their genotype, which is what the letters represent, ratio is 1 to 0 or 100%, big T, little t. The phenotype ratio, and phenotype indicates the physical appearance, is also 1 to 0 or 100% tall plants. So looking at another scenario, if we take that first generation of hybrid pea plants that are big T, little t, and we cross that one with another of the same genotype, hybrid, heterozygous, big T, little t, now I want to separate the gametes, and in this case we have from this parent one uppercase or big T, and one little t, and the same thing with the second parent. There is one gamete with a big T and one with a little t. So now when I get ready to set up my Punnett square, I'm going to need a 2 by 2 Punnett square. So the number of gametes tells us how many boxes to put in our Punnett square. So we're going to put the gametes for one parent on this side and the gametes for the other parent on this side. And when we put the gametes together, we're showing fertilization. And all the possible combinations of fertilization is what happens when we put the alleles together and determine what are the possible outcomes. Not necessarily actual outcomes, but possible outcomes. So now if we look at our Punnett square, we have a genotype ratio of 1 to 2 to 1. So let's look at that. We have 1 with big T, little t, 2 big T, little t's, and 1 little t, little t. So we have 3 different genotypes, and we have in a possibility of 100% outcomes, 1 might come out homozygous tall, 1 might come out homozygous short, but 50% possibility of heterozygous big T, little t. The phenotype ratio then, if I can get this pen to work, phenotype ratio physical, what does it look like? We have 3 to 1. We have, let me mark this with a color, tall plant, tall plant, tall plant, 3 of those, and 1 short plant. So our phenotype ratio is 3 to 1. Looking ahead to another example, let's take a look at seed color. One of Mendel's 7 pea plant traits that he studied was seed color. And if you look in your textbook, you'll see that in seed color, yellow is dominant to green. Green seeds being the recessive. It does not matter what letters you use for your Punnett squares. I prefer to use letters in which the uppercase and lowercase are distinctly different, or I can at least make them distinctly different. So let's start with the parent generation, where we have purebred yellow. So we're going to use uppercase Y, and our homozygous, and we're going to cross this parent with purebred green seed plants, and we're going to use lowercase y to represent the recessive. So big Y, yellow, little y, green. So put the gametes together. The gametes are sex cells. We only have 1 of each allele possible from each parent. And so again, our F1 generation is going to be 100% heterozygous. That is, the genotype has non-identical alleles, 1 big Y, 1 little y. This is heterozygous. Or hybrid. We have purebred or hybrid where we have non-identical alleles. And so big Y, little y is the genotype, and the ratio is 1 to 0, and yellow is the phenotype, what it looks like, phenotype. If we wanted to cross the F1 generation seed colors, we would have the F1 big Y little y crossed with big Y little y. And now the gametes, the sex cells, only have one allele from each parent. So one gamete might be this, and another might be that. And same thing with this parent, one sex cell with a capital Y and one with a lowercase y. And so again, we see that the number of boxes in our Punnett square is going to be a 2 by 2. Put the gametes from one parent here and the other parent right here. Show fertilization, and we see, just like we saw in the previous example, we have a 1 to 2 to 1 genotype ratio. One to 2 to 1, and a phenotype ratio. Three to 1. So let's take this one step further. And let's say we have a parent generation where we have the yellow seed plant, and we have, the question is are these plants homozygous or heterozygous? In other words, is the parent purebred yellow, or is it a hybrid or heterozygous yellow? Because when we look at the seeds, we see that this plant is producing yellow seeds. But the question is this plant purebred or hybrid? So what do we need to do? So we can do to test for hybrid or purebred, in other words, big Y, or hybrid, big Y, little y, we do what's called a test cross, or sometimes called a back cross. Cross the plant in question with a recessive plant. And then observe the offspring phenotype ratio. So for example, we said if we have a parent that is purebred, big Y, and we're going to cross it with the recessive, little y, we know from our previous example that all of the offspring are going to be, here's our gametes, all of the offspring are going to be 100%, here's our F1, yellow. So we can observe that all of the offspring, 100%, are yellow. If in fact our parent was not a purebred, but in fact was a hybrid, then we cross it with the recessive, our test cross, and now when we look at the gametes, this parent would either have the big Y or a little y, and again we know that this parent only offers a little y, so in our Punnett square we need a 1 by 2 box, and that we're going to put the gamete for this parent right here, and the gametes for the other parent right here, and show fertilization. And so when we put those together, we now see that there's a 50% chance of offspring being yellow and 50% green, because they have both the recessive alleles. So this is how we can determine whether a plant is homozygous or heterozygous phenotype, by doing a test cross with the recessive trait.